in the environment all the time. But at any rate, this can cause real problems. They're suggesting that the risk of contracting life-threatening diseases in this country, diseases such as diphtheria, polio, whooping cough, measles, etc., that the risk is very low. Well, that's only true if people are immunized. If everybody around you is immunized, you're not likely to pick up a pathogen from anybody around you because they are protected. But when that protection begins to wane, that's when we have problems. So most of the U.S. population is immunized. This is showing if you have a situation where you have a lack of herd immunity, or you have only 2 individuals in that population who are immunized, or excuse me, most of the people are not immunized, 2 people are infected, and then pretty soon the entire population can be infected because nobody is protected. And that's what happens when immunization is not applied. So if the majority of individuals choose not to be immunized, it impacts everybody. And it can impact some people much more severely than others. So the elderly may contract diseases that will kill them. The very young might contract diseases that will kill them. So then it becomes questionable, do you have the choice to put somebody else's life at risk just because you don't happen to like needles? So the CDC tries very hard to get people to be immunized so that we can maintain what's called community immunity or herd immunity. You have to have a critical proportion of your population protected. If they're protected, then infection is not likely to spread rapidly. And again, we talked about what kinds of things are we immunized against. Diseases that are likely to spread rapidly, or the agents causing them are likely to spread rapidly and affect a large number of people. And if the risk of contracting the disease outweighs the risk of immunizing. The CDC monitors that. They're not recommending that people be immunized against the common cold. It's not dangerous. But the diseases they want people to be immunized against are dangerous. We just don't recognize that in this country. Well, okay, so one of the risks that was mentioned in association with immunization was hypersensitivity. In actuality, immunization is not the most common cause of hypersensitivity reactions. There's lots of other things that come into play. But understanding hypersensitivity, even a little bit, requires that you have some understanding of how the immune system works. So it is a risk, as I said, but not a major one. So what is it? Hypersensitivity is an abnormal physiological state where an immune reaction, or probably more than one immune reaction, causes tissue damage or malfunction such that the body is damaged in some way. It may be life-threatening, it may not, it depends on the degree of severity. More commonly, we refer to these as allergic reactions. People are more familiar with the term allergic reactions, but hypersensitivity reactions are allergic reactions. They're immune responses, but they're immune responses out of control. So there are ways to categorize these. Like everything we do in here, we present some basic information and then divide it into categories. This can get out of hand, but hypersensitivity reactions are categorized as immediate versus delayed. If they're immediate hypersensitivity reactions, they involve B cells and humoral immunity. So B cells give rise to clones, plasma cells that release antibody into the circulation. B cells also have antibody on their surfaces. They're going to be involved in these immediate hypersensitivity reactions. Delayed hypersensitivity reactions involve T cells. So it's a cell-mediated immune response or cellular immunity response. T cells, remember, don't produce antibodies, but they do produce substances called cytokines. The other thing about these is typically immediate hypersensitivity reactions can occur within minutes of exposure to the antigen, or you might call it the allergen. On the other hand, they can persist if the hypersensitivity is to something that you have eaten and it stays within the GI tract. So it can persist. It can happen very quickly, but it can persist. Delayed hypersensitivity reactions typically don't occur until 24 to 48 hours after exposure. It takes time for the T cells to become activated and then to cause some kind of damage. Well, okay, so we have immediate hypersensitivity and we have delayed, but the immediate are also divided into subcategories. So we have immediate hypersensitivity divided into type 1, type 2, type 3. And I'm not sure if those numbers are supposed to be Roman numerals or not. So here they're not. Type 1 hypersensitivity. So these are the most common, and they involve antibodies in the isotype IgE, and they involve cells called mast cells. The mast cells have granules inside of them that have high levels of histamine, and when antibodies bind to antigen, the antibodies are stuck on the surface of the mast cells. Antibody binding antigen causes the mast cells to degranulate, and they release histamine into the environment. Here you can see it's not just histamine, but you see, let's turn this off so you can see this better. Sorry, we got that part. So here you can see the antibody, IgE, the antigen binds, and then this is showing these granules coming out of the mast cell. And again, it's histamine and other kinds of substances. This also says mast cell or basophils. Some references say that basophils are the same thing as mast cells. Mast cells are in the tissue spaces, basophils are in the bloodstream. There are very few very few basophils in the circulation because most of them leave. Other references I've seen said, yeah, we're not sure they're the same thing. But anyway, these have granules that contain lots of histamine. They also contain heparin, which we're not terribly concerned about. But they have these, they have binding sites for IgE, the antibody, and so they get stuck to those things. And when the antibodies bind antigen at their surface, again, the mast cells or basophils release their inflammatory substance, basically. So they're releasing histamine. We said histamine was a major vasodilator substance, and that it also increases capillary permeability. Well, so type 1 hypersensitivity can be localized, or it can be body-wide. These are kind of extremes at two ends of a range of reactions, because certainly there are things in between. If the reaction is very localized, type 1 hypersensitivity reaction that involves itchy eyes, runny nose, probably red, watery eyes, that is considered an atopic allergy or atopy. It's irritating, but typically not life-threatening. If you involve the airways and you release histamine, histamine can cause fluid to collect. Remember, histamine causes fluid to leave blood vessels and go into the tissue spaces, so it can cause swelling. Well, if that swelling happens to your tongue, if your tongue swells up, it can get in the way of breathing. But if it's your throat that's swelling up, that can get in the way of breathing. So it can become more serious, and then you have people with asthma. So again, we're getting an increased blood flow to the area. We've got redness, we've got swelling, there's an increase in temperature, but it's mostly the swelling that's problematic. This can be brought about by a wide variety of factors that we consider allergens. So pollen grains, fungus spores, dust mites, cat hair, lots of things that get into the air, and they irritate people. And typically you would take an antihistamine for this. You can block the effects of histamine and what histamine does. At the other end of the scale is anaphylaxis. So anaphylaxis is, again, a type 1 hypersensitivity reaction, but it involves the release of histamine body-wide. And I think I said to you guys last time, the body has something like 70,000 miles of blood vessels. Most of those are capillaries, and most of the time, most capillaries are closed. During an anaphylactic reaction, capillaries throughout the body are open, and blood goes into those capillaries. The result of that is there's not enough blood getting into the major venules to go back to the heart, and you begin to suffer from circulatory shock due to low venous return, and that's called anaphylactic shock. And it can be life-threatening in a very short period of time. So here again, it's telling you about the 70,000 miles of blood vessels. During an anaphylactic reaction, those vessels open up, blood goes into the capillaries, it's pooling, and because you're not getting enough blood back to the heart can't pump blood to the tissues. The tissues can't get the oxygen they need, they can't get the nutrient they need to continue function. And some of the tissues that are immediately affected are the arterioles that would have to contract, the walls would have to close to keep the blood from going into the capillaries. Well, if the arterioles, if the smooth muscle in the arterial walls can't get oxygen, they can't make ATP, they can't contract. And the more they relax, the more blood goes into the capillary, and it's a vicious cycle. It gets worse and worse. It's potentially life-threatening within a matter of minutes. Again, circulatory shock occurs when tissue is not getting an oxygen supply, not getting a nutrient supply, and the tissues actually begin to die. It's critical tissues. This can be life-threatening very, very quickly. And so typically what people are supposed to do, if they know they have an anaphylactic response to something like bee venom, they get stung by a bee, they have to administer epinephrine very quickly. And so they carry these EpiPens and inject themselves, which is not pleasant, but the alternative is dying, so you want to do it. As I said, those are kind of extremes. Atopic allergy on the one hand, which is just irritating, and anaphylactic reactions on the other hand, which is life-threatening within a matter of minutes. There's a whole range of different types of reactions in between. As I said, asthma involves the airways. Oftentimes the itching can be localized just in the eyes and the nose, but sometimes it's all over the surface of the skin. So if somebody has hives, that's a type 1 hypersensitivity reaction. And as I said, if it's a reaction to something you've eaten, as long as that material is in the gut, then the reaction can continue. Immediate reactions are not within a matter of minutes and then the effects go away. It can persist for quite a long time, or reoccur several days in a row. So that's type 1. Again, type 1 involves IgE, mast cells, and the release of histamine. The type 2 hypersensitivity reactions involve IgG, sometimes IgM, and complement. So IgG can go places where IgM can't. The IgM antibodies are quite large, but both of them can initiate the complement cascade, and then we know what complement factors do. They interact in sequence. If they're interacting on the surface of cell membranes, they can cause holes in those membranes, so they can damage tissue cells. They can also cause opsonization, which causes phagocytes to then go after cells. An example of a type 2 hypersensitivity reaction is called hemolytic disease of the newborn. And this is also sometimes called a cytotoxic response, but that's not a good name because there's really no toxin associated with this. Hemolytic disease of the newborn occurs when you have a mother that is Rh negative carrying a fetus that is Rh positive. Now, typically, mother and fetal blood don't cross, and the mother should not produce anti-Rh antibodies. She has the capability of doing that. She usually won't. But sometimes during the birth process, there can be an exchange of blood, and she can be stimulated to produce anti-Rh antibodies. So it's usually the second or later pregnancy where she's carrying an Rh positive fetus. She can produce Rh antibodies. If they're IgM, they can't cross the placenta. But if they're IgG, they can. They'll cross the placenta, they bind to the fetal red blood cells, initiate the complement cascade at the surface of those cells, and destroy them. So, again, an Rh negative mother carrying a fetus that's Rh positive. And this happens fairly frequently because people don't usually ask one another what their blood types are before they engage in activities that can result in the formation of offspring. Anyway, so it's IgG, and again, complement. Now, our cell surfaces do have anti-complement factors. We have ways to block complement, but apparently they're inadequate on the surface of fetal red blood cells, and so the result is typically quite a lot of destruction of fetal red blood cells, and if you destroy enough of them, then the fetus can actually die. So we know that complement can cause holes in cell membrane, but complement can also cause opsonization and attract phagocytes, so they can come in there. Phagocytes not only consume cells, but they can also kill cells. The natural killer cells can be attracted, they can cause damage. So there's a lot of things that can go on. One way that this situation can be avoided is if a woman is seeing an obstetrician prior to birth, typically she will be given injections of RhoGAM. This is a trade name for anti-Rh antibody. So she's given pre-made anti-Rh antibodies, and if any Rh antigen gets into her system, it'll bind that antibody and she won't be stimulated to produce them. And typically, if you're Rh negative, typically at the end when the first infant is born, you get one RhoGAM shot, and then if you get pregnant again, same individual, Rh positive dad, offspring is likely to be Rh positive, actually going to be Rh positive, even if they're heterozygous, they're still going to produce Rh. And so then you get 3 injections of RhoGAM during pregnancy. At least that's how it was. Is it still the same way? I haven't done this for a while. Being Rh negative and all. anyway. This type 2 hypersensitivity is also involved when you have transfused red blood cells. Again, you can have antibodies that are anti-A or anti-B within the recipient system. If they have those antibodies, they will bind to the red blood cells and then activate complement, which will cause holes in those cell membranes and destroy them. You don't want to give somebody transfused blood of the wrong type, because they're very likely to have antibodies against it, and they'll just destroy those red blood cells. There is a type 3 hypersensitivity. We're still talking immediate. Type 3 also involves IgG and IgM and complement. Sometimes this is referred to as immune complex disease, where we're complexing antibody to antigen and then activating the complement cascade. This one is not typically associated with blood cells, so it's not happening on the surface of blood cells. But again, it can be either localized or it can be body-wide. So if you have an immune complex disease and it's body-wide, it's called serum sickness. This used to be fairly common when people used large animals such as horses and goats to produce antibody that then was given to humans. This happened during wartime. They would inoculate horses with the tetanus toxin. The horse would produce anti-tetanus antibodies, and then the antibodies could be given to humans to prevent them from getting tetanus. And it was okay the first time. But then if they were given that same horse antibody a second time, the person would produce antibodies against the horse antibodies, anti-isotype antibodies, and then that would cause the damage. So these can form complexes throughout the body and cause quite a lot of tissue damage and potentially deadly reactions. If it was a localized reaction, type 3, it's called an Arthus reaction. And this is something that probably a lot of people have experienced in a very mild form. If you get a tetanus toxoid booster, and now it's usually tetanus and diphtheria comes out, it doesn't hurt when the injection is given because the syringes are very tiny and the people that are applying them are very talented at what they do. But the next day, it hurts. It's swollen, it's warm to the touch, it's red, and that's a localized type 3 hypersensitivity reaction. So type 4 is delayed. So we have type 1, 2, and 3, those are all immediate hypersensitivity. Type 4 is delayed hypersensitivity, and again, this is going to involve the cell-mediated immune response. Probably the one that's best known or best recognized, something that a lot of people have some experience with, is your reaction to poison oak or poison ivy. If you haven't experienced it yourself, you've probably seen somebody experience this. Sometimes people don't even actually have to come in contact with the plant because the plant oils can be aerosolized. If somebody's burning it and the oils get into the smoke, they get into the air, people can breathe that in and they can have this delayed hypersensitivity reaction inside their nostrils, inside their mouth, down their airways, often on the surface of the skin. What's happening here is you have oils, again, associated with the plant that become associated with skin cells. The typical reaction is contact dermatitis, where you touch the plant leaf, the oil of the plant gets on your skin cells. Your skin cells have MHC proteins, and now the T cells can recognize MHC protein plus antigen. And it's MHC class 1 because your skin cells are not blood cells. So the cells that are going to attack here are going to be the cytotoxic or killer T cells. These cells release granzyme and interferon, so they actually do begin to kill cells. People that are very allergic to poison oak will often get a blistering, red, oozing, damaged tissue on the surface. And you might think, well, they're scratching that and causing that damage. They don't have to touch it. The damage is being done by T cells. The cytotoxic T cells, they're destroying that tissue. And sometimes it can be quite severe. And again, people that are really allergic, if the aerosolized oils get into their airways, they can get allergic reactions inside the airways too. Another type of delayed hypersensitivity reaction is transplant rejection. Again, this is going to involve cytotoxic or killer T cells. And what they're doing is recognizing MHC protein that is not theirs. So they see MHC protein and they see cells that are not self, and they will attack and destroy those. This is why people that receive organ transplants typically have to be immunosuppressed for the rest of their life. And they try to match the major histocompatibility complex proteins so that they get as close a match as possible. But the T cells are very good at recognizing that which is not self and going after it. So there's a number of other conditions that involve our immune system going after our own tissues. So rheumatoid arthritis, glomerulonephritis, endocarditis, rheumatic fever, scarlet fever. These are damage to tissues in the body associated with hypersensitivity reactions. Sometimes because some toxin is associated with our tissue, and other times just because the tissue is recognized as foreign for reasons that we don't know, and then the body begins to cause damage. So immune system is primarily protective, but it's significant to recognize that in a hypersensitivity reaction, it is going to cause some damage. And so those Treg cells, the regulatory T cells, their job is to prevent hypersensitivity reactions. So their job is to turn the immune system off before this kind of thing happens. And as I said earlier, proper development of the immune system apparently is influenced by gut microbiota. There's pretty good evidence to suggest now that people need to have a very diverse gut microbiome in order to have a properly developed immune system. And we're messing with that a lot. So gut microbes also influence hormone levels. They regulate the autoimmune disease development and influence developmental aspects of the adaptive immune system. As I've mentioned, the details of this kind of thing are beyond the scope of this class, but basically, as I've mentioned before, be nice to your normal microbiome, because you need those guys. Okay, so that wraps up hypersensitivity. From here, we're going to epidemiology. And this section, in the notes that are in the notes that are online, I think it says the quickie version of epidemiology. Yeah. And I've gone through and looked back at micro textbooks now, and it's like, oh my word, a lot of the terminology has changed. So I'm really giving you a quickie version and a very simplified version of epidemiology. But considering where we are and the fact that we're kind of behind in lecture still, it's probably okay. You guys are almost overwhelmed anyway. Almost.
Way past that.
We just have, yeah. So we only have, a lab exam this week and then a final exam next week, and we've tried to get
And
through
projects
this.
due.
And take home quiz.
Yeah, we
And
have to take
semester
home quiz tonight.
project.
Yep. So
It's
what the
fine,
heck
I'm
is
fine.
epidemiology? Epidemiology is the quantitative study of the occurrence of disease. Notice quantitative is in a bright color there, so you pay attention to that. It's about numbers. We're looking at disease, but we're looking at the occurrence of disease and factors that influence frequency and distribution. So you're counting, you're collecting data. I'm going to show you guys a film next Tuesday in lab, because next Tuesday we're finished with lab, but we have a film on epidemiology. It may be a film that some of you have seen. But if you have, you should see it again and think of it in terms of epidemiology, what's going on. Because you get to see people from the CDC, Centers for Disease Control and Prevention, in action, and you get to see some of the things that they're trying to do. And it all has to do with numbers within populations. So we're talking about disease in populations as opposed to disease in individuals. So epidemiologists collect data. They analyze patterns, they look to find out what are causes of outbreaks of disease, what kinds of things are affecting the spread of those disease, and mostly they're concerned with how do we prevent disease. And again, that's disease within populations. So they're looking at the global population. This is a cornerstone of public health. Notice should be in a different color. It should be used to shape policy decisions and design evidence-based practices by identifying risk factors for disease and targets for preventative health care. Should be, not necessarily is. And it has to do with funding, and it has to do with politics, and it has to do with people making decisions based on, I'm not sure what, because it certainly is not data-driven. It's very frustrating to see what's going on relative to epidemiology in many parts of the world, and here in many instances. Yeah, oh, the victims, all of us. Well, okay, so it has changed a lot over time. New studies provide new data, and there's new ways to analyze it, and there's a lot of subcategories that come into play here, and you guys aren't going to get any of that, because this is the quickie version, and it's way simple. So, our need for epidemiology is extreme, and I might point out that this isn't something that's going to go away, because actually climate change is going to raise sea levels, and when sea levels raise, the amount of Earth that can be occupied by humans is going to decrease, and populations are going to be squished even more than they are now. And when populations are very close, when individuals are very close, it's real easy for pathogens to be passed from one to the next to the next. Immunization becomes so important, and then we have all these people that decide, no, I don't want to be immunized because some actor someplace said it wasn't good for me. What? Anyway, so there's some history here. Hippocrates, who was a Greek physician, was sometimes called the father of medicine. If you look into various texts or books that have been written about medicine, you see all these people that are named the father of something, like Leeuwenhoek and Koch and so forth. Anyway, so he was the father of medicine. He was the first person credited with recognizing a relationship, a causal relationship between occurrence of disease and environmental factors. He was one of the people who said, you know what? Maybe you shouldn't drink water out of that river after it's gone through the city. You want to drink water out of that river, do it before it goes through the city. Because when it comes out at the other side of the city, it's not nice. We're going to talk a little bit about that tonight because I did not give you guys information about water treatment, and I have to. Because I put it on the exam.
I mean, you didn't have to.
This exam? Like Thursday's
Not
exam?
anymore.
Like on Thursday?
Thursday's exam?
Thursday's exam.
Yeah, but
That's all right. We'll get through it.
alright.
It's not a big deal.
It's fine.
Okay, so I did mention Girolamo Fracastoro. He was the person that suggested that some kind of small unseeable particles were the causative agents of disease. He used the term fomite to suggest that something should be transmitting disease, and that the disease-causing particles were something we couldn't see. That was back in 1543. People didn't pay much attention to it because they didn't have microscopes. He couldn't prove that there was something you couldn't see. We now know he was absolutely right. It wasn't until von Leeuwenhoek developed microscopes and began to see microorganisms all over the place and said, hey, look at this. These things are all over the place. People began to think, maybe some of those little tiny things that you can't see with the naked eye might have to do with disease. And then, of course, we had Robert Koch, who came along and said they absolutely do. Some of them can cause disease, and et cetera, et cetera. Anyway, so the overall goal of epidemiology, as I said, it's the quantitative study of the occurrence of disease and factors influencing frequency and distribution. Why are they studying it? Because they want to prevent disease. Epidemiologists, their goal is disease prevention. Within the US, data is collected, analyzed, and distributed because they have to collect data about what's going on with diseases and then get that information out to clinics to let clinics know, for example, what vaccine should you be using against influenza this year based on evidence or data they've collected. Anyway, in this country, it goes to the Center for Disease Control and Prevention, which is the CDC. The acronym CDC is just Center for Disease Control. But what it was recognized after not too long a period of time, you really want to control disease. You have to prevent disease. And that's what immunization is for, is disease prevention, to prevent people from getting sick in the first place. Anyway, the headquarters for the CDC are in Atlanta, Georgia. As I said, I'm going to show you this film next Tuesday. You see them going into the CDC headquarters and you see people working in there. The only thing that's not accurate about the film is that they have televisions all over the place. The reason the televisions are in there is because part of the movie is soundtracks from news. And they have that going all the time. So you can see what's going on in the news. They don't actually have those televisions all over their laboratories. On the worldwide level, the World Health Organization, WHO, again, is collecting data and analyzing data and distributing information. Their headquarters are in Geneva, Switzerland. So these institutions gather and analyze data, they make decisions, and then they provide information to healthcare facilities and healthcare workers. Again, their overall goal is disease prevention. So one of the newest ones was Zika, and what should you do in response to Zika? Where should you not go? Who should be immunized, or what steps you should take? Those are the kinds of things they do. So the purposeful dissemination of misinformation and alternative facts with no basis in reality is extremely detrimental to this process of actually collecting data and making decisions. And unfortunately, this happens in this country a lot. I just saw these commercials talking about cigarettes. Well, the cigarette industry for a long time told people that doctors felt smoking cigarettes was good for you, was actually beneficial to your health. And so they promoted it. Meanwhile, the tobacco industry is making money hand over fist, and they knew full well that they were hooking people on an extremely addictive substance, and that would eventually kill them. They continued it. So dissemination about immunization is the same kind of thing. It's not functional. It's not helpful. And you see this kind of thing all over the place. Okay, well, it's good for you. What if it was? It's okay, I just saw another one. There was a very large gathering of people that honestly believe the earth is flat.
Oh, yeah.
Oh, well. Now, we're going to encounter a lot of terminology that goes along with epidemiology, and this terminology, again, it's putting things into categories. So, disease categories based on epidemiology. If a disease is endemic, endemic means that it is constantly within a population. It's always there. There may be just a few people being affected overall over time, but the level of infection tends to stay about the same. So relatively few cases, but occurring all the time. An example of an endemic disease is the common cold. It does fluctuate because in the wintertime we have more common colds because people are crushed into dwellings inside and they're breathing on each other more. In the summertime they're outside and it's harder for the viruses to get from one host to the next. But that would be an example of an endemic disease. Apparently the term endemic was coined by Hippocrates, and this had to do with diseases usually found in some places but not in others. Common cold is pretty much all over the place. Plague is endemic in California. This is something we don't typically think about. It's not endemic in human populations since it's endemic in rodent populations in California. You can't immunize rodents, you can't eliminate all the rodents, and so it's just there. There are always some rodents in the state of California that have plague. And it's important to recognize this. Again, the CDC's response to it is to post plague warnings. If they find animals that are infected with plague, let people know that there's plague in an area and tell people don't mess with those dead squirrels. Leave them alone. Don't put your tent on top of a squirrel dwelling. Another term is sporadic. If a disease is sporadic, it occurs in small, localized, unpredictable outbreaks. An example of sporadic might be malaria in California. We have Anopheles mosquitoes here. Malaria is certainly not endemic to California, but if somebody comes to California carrying the plasmodium within their bloodstream, and that is picked up by a mosquito, and then the mosquito survives and goes off and bites somebody else, you can have a little outbreak of malaria. And this has happened in California numerous times. Legionnaire's disease is another one that is typically considered sporadic. It's associated with water. Oftentimes the sprayers that mist your vegetables in the supermarket are probably more likely the ones that mist you in the summertime when you're going to the fairs and whatnot, and they have them up above doorways and you go underneath. They're supposed to cool you off, but you wonder, what's that water source? How clean is it? And do we have Legionella pneumophila in the water? If a disease is epidemic, that means the number of cases is significantly above an expected background level. Well, you can't have an expected background level unless you're monitoring. And again, that's what epidemiologists are doing, is collecting data and monitoring what's the level of disease to know if you have more incidence or greater incidence of disease, maybe it's becoming epidemic. Again, Hippocrates used the term epidemic for diseases that are seen at some times but not at others, but they would have been seen, a bunch of people have them, you'd have an outbreak of disease, and then it might disappear for a while and then come back. Significantly above an expected background level. And when you have a disease that is epidemic, and that epidemic is affecting people in multiple nations on more than one continent, that's called pandemic. There are a number of diseases that have been pandemic in the past. Plague was one that spread from various regions of the world because it was carried by rats that got off ships when they came to port. Then they would go and infect other rats in cities and so forth. Smallpox became pandemic, partly because Europeans came here and proceeded to give blankets and whatnot to the native people in this country, who then got smallpox and died from it. They also got measles and died from that. And Ebola has become pandemic to some extent. Influenza is pandemic. So those are some terms that apply. You need to be familiar with those for test-taking purposes. Then we can look at factors that influence the prevalence of disease. So if you're an epidemiologist, you want to know, well, what kinds of things you have to take into account. If you have a disease and you want to know how is that disease spreading, you need to know what are the reservoirs involved and what is the mode of transmission. For epidemiology, the term reservoir doesn't necessarily refer to a body of water. On the other hand, it can. The term reservoir applies to the sum total of all potential sources for a disease-causing agent. So a reservoir could be soil, it could be water, it could be plants, it could be animals. Consequently, reservoirs can be categorized as living or non-living. I think non-living are going to come here first. So non-living reservoirs. When you think about where can microorganisms exist? Well, water is an excellent non-living reservoir. Lots of microorganisms can survive in water, especially bacteria. Bacteria get washed into water. They're coming from the surfaces of plants. They're coming from soil. They're coming from wastewater treatment facilities. They're coming from people just depositing their excrement in the water. So they can get into water. They may not be able to reproduce there, but they can survive for a long time. Some of them can reproduce there, and so there are certain pathogens typically associated with water, like the vibrios that can cause cholera, some pseudomonads, and so forth. And here you see various examples of water in different stages of pollution. This is a serious problem. Another potential reservoir is air. Again, microbes get into air. Anytime the wind blows, you see these dust devils that are picking up soil and flinging it into the air. Microbes are going along with that. Microbes have been found very high up. They're up in the jet stream. They're traveling. They travel huge distances. So in this class, when we do our semester project, it's not unusual for people to find organisms that were identified initially in Antarctica or in China or someplace like that. And people go, wow, how did they get from California to China? Well, maybe they got from China to here, but they just travel in the air. They don't reproduce in the air, but they can survive there for long periods of time and get carried all over the place. When humans cough or sneeze, they release little packets that are called droplet nuclei. These are mucus and water and microorganisms associated. The water very quickly evaporates, and then the mucus, which is sticky, kind of ties the organisms together in these little particles. These droplet nuclei can be expelled, and then again, the organisms can survive for a long period of time. Yeah, nice. So when we talk about transmission, you can argue about is transmission direct or indirect. Well, it depends perhaps on how close you are to the release of the droplet nuclei. If somebody sneezes on you, that's pretty direct transmission. But if they sneeze into a room and they go out of the room and you come in within the next few minutes, those droplet nuclei are still hanging around in there. So is that direct transmission? Kind of, sort of. It depends on which book you're looking at. Soil is another excellent reservoir. There are lots of microorganisms, particularly bacteria, that live in soil naturally. That's their normal habitat. And if they get out of the soil and get into an organism where they might cause disease, sometimes it's almost an accident. Kind of interesting, I read an article about Bacillus anthracis. Apparently Bacillus anthracis is maintained not just loose in the soil, but inside earthworms that live in the soil. So there can be organisms that live in the soil that are carrying these microbes around with them, and they're maintaining the populations of these things. Again, lots of nutrient available, lots of things can live in the soil. Or play in the soil. Look, there's a person getting their immune system stimulated. Lots of organisms besides bacteria, fungi, protozoa, etc. live in the soil. Another potential reservoir is food. And how good a reservoir food is depends primarily on how much exposure it has to air where microbes might get into it, and then what temperature is it maintained at. We know that food has lots of nutrients. So if you prepare food and then let them sit at room temperature, a lot of organisms that are disease-causing agents are mesophiles. And so that's where they're going to survive and grow. And you may not necessarily know they're there. It's not always easy to recognize them. Another category of reservoirs is fomites. As I said, this term was first used by Girolamo Fracastoro, but fomites are small items, typically utensils, bedding, clothing, things that you handle, and then microorganisms can get onto those things, and then somebody else comes and picks them up, and they can get the microbe. Supposedly money in this country is less able to transfer pathogenic organisms because of the heavy metals. So silver and copper tend to inhibit microbial growth. Does it necessarily kill them on coins? Not right away. So you can take coins and put them on a nutrient auger plate and you will get some growth. Not as much as you get from a dollar bill though. Paper money has lots of things. So again, bacteria don't typically reproduce on fomites, but they can be carried around on these and persist for a long period of time. Depends on the organism. Now, another term that we encounter when we're talking about non-living reservoirs is the term vehicle. If you look this up, if you look up the term vehicle, probably what you'll see is automobile, motorcycle, plane, boat, etc. Yeah, those are vehicles. But that's not what we're talking about. In epidemiology, a vehicle is a non-living factor that is involved in disease transmission, or that is technically it's involved in the transmission of organisms that can cause disease. I wrote this thing, and then as I go back and look at it, and I think you see this a lot in public health documents. They talk about disease transmission, and they talk about disease being passed from individual to individual. Remember, it's the disease-causing agent that is transmitted, not the disease. And whether you get disease or not depends on your immune system, how much pathogen you're exposed to, and a lot of other factors. But it's not really disease that's transmitted, it's the disease-causing agent, even though I have this written here and undoubtedly in the notes. So again, vehicles are non-living, and they're involved in the transmission of disease-causing agents from person to person. So a lot of these non-living reservoirs can be vehicles. Water, air, for sure. They move things around, and people can get in and out of them. So then we look at living reservoirs. First comes to mind is humans, because we are reservoirs. We're living organisms. We can harbor quite a number of microorganisms, and sometimes they are causing disease in us, and sometimes they are not. There are quite a number of bacteria and other types of organisms that cause disease in humans and not in other types of animals, so they're restricted to the human population. This was true of smallpox, which was why the World Health Organization was able to, first of all, they recognized this, that the virus that caused smallpox was restricted to the human reservoir. They decided that they would immunize the entire world population. So the World Health Organization set out to do exactly that. I'm not sure exactly how it was accomplished, but a lot of manpower, a lot of people involved. And in 1979, they declared smallpox eradicated. It's gone. Unfortunately, the smallpox virus has been maintained in numerous facilities, and Now, because military personnel are being immunized against smallpox, there's a potential for the smallpox virus to reemerge as an infectious agent. Sometimes humans are reservoirs and they don't show any disease symptoms. This is true because a lot of us have organisms that live in our gut that are potentially pathogenic to somebody else. Your E. coli are fine for you, but they might cause disease in somebody else. You can have Neisseria living in your pharynx that are fine for you, but they might cause disease in somebody else. And apparently there was one particular individual, her name was Mary Malone. She came to be known as Typhoid Mary because she was carrying Salmonella. Now it's recognized as Salmonella enterica, subspecies typhi, but it was the organism that causes typhoid. And she was working in food handling, food preparation, and was not real big on hygiene. Perhaps the facilities were not such that they allowed her to concern herself with hygiene. And so she infected a lot of people. She didn't believe that she was responsible for causing any kind of disease in somebody else because she didn't have any symptoms herself. She didn't have typhoid, she never had typhoid, but she was passing it to other individuals. And eventually this person was incarcerated. She was incarcerated for a period of time and then released. Was not supposed to go back to the food handling business, she did, and ultimately was locked up. She was put on some island and had to live the rest of her life there. She was very unhappy about this. Yeah, not good. Now, non-human animals can also be reservoirs, not surprisingly. Typically, we think of non-human animals, we think mammals, birds, reptiles, but remember, non-human animals also include the vectors that we talked about in laboratory, the ticks, the lice, the fleas, etc. So these can be important reservoirs. A lot of those organisms can carry microbes that might be passed to us. There's a term that applies to diseases that are normally associated with non-human animals, but then the disease-causing agents can be passed to humans. Those kinds of diseases are called zoonoses. If it's one, it's a zoonosis. But if there are multiple, they're zoonoses. Anyway, there's quite a number of diseases that we now recognize are truly zoonoses, and they're getting into human population accidentally. Plague is one of these. It's normally a rodent. Well, as I said, it's endemic in rodent populations in California. It's kind of an accident that plague gets into the human population. Rabies is another one. It's endemic in bats and foxes and raccoons and so forth. I'm thinking of just not coming into my mind. It's like, no. Meningitis. Meningitis is another one that is a zoonosis. Influenza. Anyway, so arthropods are non-human animals, but arthropods play a dual role in terms of epidemiology because they're reservoirs, they're also vectors. The term vector applies to a living agent that can be involved in disease transmission. Again, what they're doing is they're involved in transmitting disease-causing agents, not transmitting disease. But ticks and fleas and lice and so forth, they're reservoirs and they are vectors. Okay, so one factor influencing the spread or prevalence of disease was reservoirs. Another factor is mode of transmission. How are diseases transmitted? This is a section where there's a lot of change in terms of categorizing. Again, this is a real simplified view. So I'm going to say that disease transmission or transmission of disease-causing agents is either direct or indirect. But the terms may not mean exactly what you might think. So, yeah, more complex. Direct transmission, it's that occurs between a susceptible host and a living reservoir. So living to living is direct transmission. It can be human to human, but it doesn't have to be. It could be mosquito to human. So if you're bitten by a mosquito that's carrying the Anopheles, the Anopheles mosquito is carrying Plasmodium that causes malaria, that's direct transmission because it's living to living. If you're bitten by a rabid dog, that's direct transmission. If you're licked in the face by a rabid dog, that's direct transmission. If you eat pork that contains Trichinella spiralis and it's not quite dead because you didn't cook it enough, that's direct transmission. It's from living to living. Kind of gruesome. How long does it have to be cooked? Long enough to kill those things. So syphilis is human to human, definitely direct transmission. Plague, rabies, typically from animal to human, still direct transmission. And indirect transmission requires that you have a living entity and then a reservoir or vehicle that's non-living and then back to a living entity. That makes it indirect. So the disease-causing agent is passing from a susceptible host or passing from an infected individual to a susceptible host by means of a vehicle previously in contact with a reservoir. But the reservoir can be the vehicle, so it doesn't necessarily mean that those are separate. So these people that are getting into water, they're getting into water, if they drink that water and they pick up Vibrio cholerae, that Vibrio was released by somebody else who put it in the water. That's indirect transmission. If you step on a nail and you get Clostridium tetani into your tissue, the Clostridium tetani probably came from the soil, a non-living reservoir, and then it got on the nail, a non-living vehicle, and got into your skin. That's indirect transmission. Yeah, but if you look in our textbook, it's much more complicated. So you can have contact transmission, which is something like direct person-person, such as the transmission of Treponema pallidum, causative agent of syphilis, but it can also be indirect or droplet, and it's still contact transmission. It's like, it's getting more complex. Transmission can be vehicle transmission, can be direct contact transmission, can be indirect contact transmission, which this is what I would have just called indirect transmission. Oh, those are nasty syringes there.
Yep.
And they put droplet transmission as a separate category. So again, this is coming from our textbook now. Droplet transmission is a third type of contact transmission where the susceptible host is exposed to a pathogen within droplet nuclei. Only certain types of pathogens apparently are transmitted this way. And then if you have vehicle transmission, this is going to involve water, food, blood, drugs, etc. You're still going to see overlap between these categories, and so it gets really confusing. I think for test purposes, we'll stick with the simple. Direct is between a living individual that's living and infected coming in contact with another individual that's living and passing the disease-causing agent that way, from living to living. If it's indirect, there's a non-living entity in between. Part of the reason I don't want to get into as much detail is because you start looking in different places, and these categories are not the same. So the CDC has a different set, and some of the others' textbook has a different set. So we'll just keep it simple. Nevertheless, if we're trying to figure out, well, why can some things be transmitted only directly and other kinds of things can be transmitted indirectly, this has to do primarily with the sensitivity of the pathogen to exposure, or how hardy is the pathogen. And when you think about what kinds of factors pathogens might be exposed to, if they get out into the environment, they can dry, they can be exposed to temperatures, extreme heat or extreme cold. Those can influence them. They can be exposed to radiation, ultraviolet radiation, etc. And so, depending on how hardy they are, they may not be able to survive that. Typically, the ones that have to be directly transmitted are kind of weak. They don't have a good survival ability. Something like salmonella, the Salmonella enterica that can cause either typhoid or just, well, there's a lot of subspecies of Salmonella enterica. Those guys can survive in dry powdered milk for 2 years. They're just sitting there. They're not growing, but they're not dead either, which is kind of scary. So how robust is your microbe? What kind of harsh conditions can they survive? So then we're going to look at factors that influence severity of disease. Again, if you're an epidemiologist, If your job is to prevent disease, then you're gonna be looking at your population and saying, what can I do to protect my population from disease that might be coming along? So what kinds of things do we have to consider? One is acquired immunity. Host resistance, this is acquired immunity. We've talked about acquired immune system, adaptive or acquired immunity, both humoral and cell-mediated. Please don't talk during lecture. So it seems then kind of intuitive that the best thing you can do for your population is to immunize them. Assuming cost wasn't a factor, but in actuality, cost is a factor. The other thing that can be a factor is availability of vaccine. You need to immunize your entire population, but you can't get a hold of the vaccine, so what do? Nevertheless, this is a significant thing to keep in mind. Another thing that can be considered is genetic background. Some populations are more susceptible than others. I think I mentioned this earlier, but people that have sickle cell anemia are actually more resistant to malaria than people that don't. So sickle cell anemia is itself a disease that's damaging, but it's not as damaging as malaria. So people that have sickle cell disease are better able to survive infection with Plasmodium than somebody else. Beyond that, people have quite a lot of variation in their immune system simply because we have all these subpopulations of B cells. You're supposed to have very large numbers of subpopulations of B cells. Some have more than others. Some people have relatively few, and they don't have a very robust immune system because of that. Okay, so I've talked about that. Another factor to consider is the general state of your population. How old are the people? Are they well fed, or are they half starved? Is there some pre-existing disease, etc., that would make them more susceptible to some new disease that comes along? This is a major factor. Typically, very young individuals or very elderly individuals are more susceptible to disease. So if you're looking at your population, you would take those kinds of things into account. On the other hand, this may be very difficult to change. It's going to be tough to change the general state of your population. Assuming money wasn't a factor, well, you'd want them to be in the best health possible. So you'd want to treat them and try to prevent disease if you could. You'd also want to make sure they were well fed, well housed, well cared for, etc. As opposed to what we seem to be headed for in this country, which is exactly the opposite. Another factor is intensity of exposure, and this can be intensity in both time or either time or space. If you are living in a village where your only water supply is from a single well, and everybody in the village is taking water out of that well, and that well suddenly becomes contaminated with Vibrio cholerae, it's very likely that the entire village is going to be infected with Vibrio cholerae, which is the causing agent of cholera. On the other hand, if you were somebody living in this village, maybe you had been exposed to Vibrio cholerae all your life, low levels of it, so you were actually immune to it. Somebody just passing through takes one drink out of the well, and they get cholera. So there's a lot of variation here in terms of intensity of exposure. Don't go to the local well lemonade stand. Wonder about those. Where are they getting that water for their lemonade? Okay, another factor that can influence prevalence is cultural practices, basically the habits of the people. What are people doing? So if they're using river water as basically their toilet, and they're also using that river water for bathing and for drinking, they're likely to be exposed to pathogens much more so than people that live in this country. On the other hand, this is one of those areas where I like to ask people questions about what we do as a society. Some of you are probably familiar with laws that have to do with wastewater in this country. So if you're establishing a residence, you have to put in facilities to deal with wastewater. You either put in a septic system with septic lines, or you connect to a sewer system. And the sewer then takes your water away and it goes to a wastewater treatment facility. And yet, how many of you have seen disposable diapers deposited everywhere? They're beside the bench at the park. They're sitting beside the sidewalk just randomly. They're outside the back door of some supermarket. And it's like, what the heck? as a nation, as a society, are we nuts? Why do we allow disposable diapers when we have all these laws about what can be done with human action? It's a mystery to me. And yet, because they're so easy to use, it would be really hard to get rid of them. Really difficult to get rid of them. They're not particularly nice for babies. If you had an infant, and if you could get diaper service where they deliver sterile diapers to your door, you're so much better off. The only problem with them is they leak. They are kind of leaky, even if plastic.
There's new ones that are out there. There's like a soft diaper movement going on.
Yeah, and there should be, because this is crazy. I mean, it's just
for my kid, and it doesn't really leak as bad as it used to.
Oh, well, that's good to know. Yeah, when I used them on my kids, and it was like, oh my goodness. But the good news was you knew when those things were wet. And so you see the commercials that say, here, this little dude, you can walk around for hours and you won't ever know. It's like, yeah, but what's it doing to the little dude? That's not good. That's not good. Yeah, so we have laws, got to be connected to sewage, but look at those disposable diapers. There they are out in the environment, and of course they get scooped up and put into landfills, what we call sanitary landfills. And I really wonder about the sanitation of our sanitary landfills, because that's not the only thing that's in there that is human waste. Pretty scary stuff. Okay, another factor is virulence of the pathogen, and this is kind of lead us into our next topic, which is mechanisms of pathogenicity. So virulence it's kind of a tough term to define. It's a degree of pathogenicity. Typically, if a microbe is said to be a virulent pathogen, that means it's very likely to cause disease. We'll come back to virulence in a bit here. So if you're an epidemiologist and you want to control disease, actually you want to prevent disease if possible, so you want to think about how do you do that? What's the best way you can control epidemics? One of the first things you can do is increase host resistance. So if money was not a factor, what would you do to increase host resistance? I mean, you could feed people better, maybe. You're not going to change the age of the elderly, make them young. You can wait for the really young to get older and then they'll be more resistant. But what can you do in between? Immunize. You can immunize, and that's what the CDC tries to do, and that's why they have those lists of immunizations that people should have so that they can be protected against disease-causing agents. Of course, the other thing you can do to help increase host resistance is educate, and that's really tough to do also, because you have to educate people about immunization first, but you can also educate people about, like I said, the plague notices. When the public health officials recognize that there's plague in an area, they will post notices that tell people don't mess around with dead squirrels, don't put your tent on top of the rodent burrows, that kind of thing. They're trying to educate people. So both immunization and education could increase host resistance. Again, assuming cost is not a factor. Provide immunization, educate. There's some educational warnings. One has to do with paralytic shellfish poisoning, telling you don't take those shellfish because they're toxic at this time of year. This one's talking about ticks, they might be in the area, and the other one's a plague warning. But how easy would it be to educate people about disposable diapers? It'll be interesting to see. Another thing you can do is decrease exposure to reservoirs and or vectors. So how do you do that? Well, you can eliminate vectors in some cases. So we do have vector control programs in California. We have a lot of water in California, so we try to control mosquito vectors, and that's an ongoing process where they're constantly trying to get rid of excess numbers of mosquitoes. As I said, you can post notices to try to prevent people from encountering the fleas that might be getting off of rodents. But you can't really eliminate the rodents, and you can't really eliminate the fleas either. So probably education comes into play here. If you want to decrease exposure, here's California's rice fields and the mosquitoes that we're trying to control. You can't immunize rodents, you can't get rid of them all, so plague is going to remain endemic. And then the third thing you can do is isolate or segregate. In a sense, this kind of overlaps keeping people away from reservoirs and vectors. But this is tough. So supposedly schools will tell you if your child's sick, don't bring them to school. How many parents do it anyway because the parents have to work and they can't afford daycare, so they dose them with medication and take them to school. And by the time they get to college, they dose themselves with medication and drag themselves to school anyway. It's like, oh yeah, we're gonna get exposed. Isolation and segregation has some rather negative connotations anyway. People don't like the idea of being segregated. But in some instances they are, in clinical settings they are. Here's some quarantine signs. A little bit more earnest about their quarantine there. Use of deadly force authorized for that quarantine in the middle. So then we can think, well, if you're trying to determine how well epidemics are actually being controlled, you need to know about these factors. So here's some other terms you need to know. Mortality rate and morbidity rate. So the morbidity rate is the number of cases of a particular disease per unit population and per time period. As I said, epidemiology is a quantitative study. So to get these kinds of numbers, how many people have been infected with a particular disease-causing agent per unit population, per time period, that's a lot of data collection. So you should be able to look something like that up. You wanted to know what's the morbidity rate of plague in New York City in 1980. You might be able to find that out. You might be able to look it up. Somebody probably keeps those records. Meanwhile, mortality rate is the number of deaths attributed to a specific disease-causing agent per unit population and time period. And the CDC publishes weekly morbidity mortality reports to let you know what's going on. So they are collecting this data and getting the information out. The MMWR weekly submission system. Here's talking about Zika and what's going on. But you can read these, they're posted online. So again, the CDC is the organization in the United States that's collecting data and putting information out for people to look at. Contains a wealth of information. It's divided up into different categories that you can look at. And as I said, their modes of transmission don't match the ones in our textbook. And that's the end of that section. Might have to stop because I don't know where the other one is. But I think, are we back on track time-wise?
Yeah.
Yeah.
Just have one left.
We have
Yeah.
one left.
So next time we'll look at mechanisms of pathogenicity, because I can't find it.
Just wait till next.
Ah well.
There it is.
Thanks, Jelena.
Yeah.
There it is.
Oh.
Started the back row.
Oh.
100 slides. Oh my god.
Oh my word.
I
We won't get through this anyway. Here it is. Pathology and mechanisms of pathogenicity. So, as I was talking about factors that influence prevalence, one of the things I mentioned was
out
virulence.
of control today.
But before we get to virulence, I want to say a couple of things about pathology, which is another way to look at disease. So if epidemiology is the quantitative study of the occurrence of disease and factors influencing distribution, etc., pathology and epidemiology has to do with populations. Pathology is the study of disease in individuals. So you're looking at what's going on with the individual. It's the study of disease dealing with etiology, which is a cause, what caused the disease, and then the pathogenesis, which is what's causing the development of disease symptoms within that individual, and then looking at anatomical and physiological changes occurring in association with that disease within an individual. So why is a pathologist interested in knowing all these things about disease? You can almost look at this list and think, well, they just want to know how awful this disease is going to be. But their overall goal, again, is disease prevention. So if they can understand the processes, then they can understand what might happen next and how do you stop that. They're trying to control disease, but they're trying to control or prevent disease within individuals rather than within populations. Some of this is review, which is fine because review is always good. There's a difference between infection and disease. So infection occurs when microorganisms are colonizing a region of the body where they don't normally inhabit. And they can get into the bloodstream accidentally. Just cleaning your teeth, you can introduce a few microorganisms into the bloodstream, but they're not normally going to stay there. Your immune system's going to get rid of them very quickly. So infection doesn't necessarily mean disease. A disease occurs when the infection is causing negative changes. So you've gotten to the point where pathogenesis is happening, changes in the anatomy and the physiology of the individual that are negative changes, that is detrimental to the overall health of the individual. Then you have disease. And because diseases vary, so we had a bunch of terms relating to disease that were epidemiological terms. Now we have to look at some disease categories that are pathology terms. If a disease is acute, that means the symptoms develop quickly and the disease doesn't last a very long time. So maybe you get something and you're sick with it for maybe a couple of weeks and then it just goes away. Common cold is a good example of an acute type of an infection or disease that would be considered acute. If a disease is chronic, then the symptoms develop slowly, oftentimes over a number of months, sometimes over a number of years, and the symptoms persist for a very long period of time. Something like mononucleosis is a good example of a chronic disease where the symptoms kind of creep up on people. They may not recognize they're sick, and eventually it becomes worse and worse, and then they seek a physician's help. On the other hand, some of these diseases that might be chronic can also be latent. Tuberculosis might be a disease that is considered chronic because it takes a long time for tuberculosis to develop, but people can have symptoms from tuberculosis and then their immune system can control the mycobacterium that's causing tuberculosis for a little while. You can kind of wall them off in the lungs and they can stay inactive for a long period of time. When they're inactive, they're in a latent state. So Mycobacterium tuberculosis can cause latent disease. It also causes chronic disease. Some types of viruses also can do this, where they can be latent. We talked about human herpes viruses. They're latent, they're inactive for long periods of time. So acute, chronic, latent, those are terms you need to know. We can also have some terms that apply to either pathogens or their products in the bloodstream. These are terms many of you are probably familiar with. If you have bacteremia, it means there's bacteria in the bloodstream. They don't belong there. That's not a place where you would normally find microorganisms, specifically bacteria. So if they're there, that's considered an infection, even though it may be a very short-lived kind of a thing. Septicemia is when the bacteria are in the bloodstream, they're reproducing and they're causing disease symptoms. And I have to qualify that because although if you look up the term septicemia, that's probably the definition you'll get. Septicemia is now also applied to certain types of fungi. So you can get septicemia with candida infection, with cryptococcus infection, possibly other types of fungi that can get into the bloodstream. That's still considered septicemia. And then toxemia, that's when toxins are in the bloodstream. Toxemia can be caused by things other than bacterial toxins, but because we're talking primarily about microorganisms, usually toxemia for us would be bacterial toxins in the bloodstream. Diphtheria is due to toxemia. The disease is actually due to toxins in the bloodstream. Rheumatic fever, scarlet fever, the disease is due to toxins in the bloodstream and our reaction to those toxins, which is hypersensitivity. And then if you have viruses in the bloodstream, that's viremia. More terms. So then we get to mechanisms of pathogenicity. So again, pathogenicity is the ability of specific types of organisms to infect and cause disease symptoms in some other type of organism. Pathogens, they're organisms that are going to cause disease symptoms. They're going to cause harm. And if they're virulent, if pathogens are virulent, as I said, virulence is a degree of pathogenicity. Typically, if an organism is considered virulent, that means it's very likely to cause disease. So virulence in bacteria can be due to genetic variation. One thing about bacteria, there are lots of different strains of them, and many of them are relatively non-pathogenic. Then you'll get some specific strain that might be highly pathogenic. You look in the back of the lecture syllabus where it's talking about the dark side of E. coli. There are all these different strains of E. coli, and some of them can cause very severe disease symptoms, whereas others not at all. Typically, we don't think about the E. coli we work with in this laboratory as being particularly dangerous, but some strains can be. And it can be due to genetic variation, difference between Bacillus anthracis and Bacillus cereus, plasmids, as far as I know. It's genes carried on plasmids. If they have the plasmids, they're pathogens. If they don't, they're not. Then we learned about bacteria that can produce toxins due to lysogenic conversion. So they're carrying viral genes, and they're expressing viral genes. Well, okay. Virulence can be expressed in terms of ID50 and LD50. These are terms, again, that have to do with how many people are infected. So well, not necessarily people, can be animals. ID50 is infectious dose. So it is the concentration of pathogen, or the amount of pathogen you have to infect 50% of a population. That means some type of pathogen is being put into a population, probably test animals, and you see how much, what's the dosage required to infect 50% of that population. Some types of bacteria, it's a very low number. Something like Shigella. One of the reasons we don't work with Shigella in the laboratories, it only takes about 200 cells to give you a severe Shigella infection. You could easily pick up 200 cells, bacterial cells, and infect yourself with those guys. The lethal dose, the LD50 is lethal dose. It's the concentration, again, cells or virions, that's going to kill 50% of a population. So how are these numbers arrived at? Using test animals in some instances, or looking at populations. What you see in the picture on the right, that's the cholera epidemic in Africa, where very large numbers of people died. So next time we'll talk about how to be a successful pathogen. So what do you have to do to be virulent? But we won't start that today. Yep, we'll just start. So you do have a quiz tonight, but the quiz is take home. Hey.
Oh my
Take home quiz, I'm just going to give it to you. So go ahead and take a break, but when you come back, I want to give you the study guide for Laboratory Exam 2. I know it's on the website. Some people have printed it, other people haven't. I want to go over it with you, so I decided I'm just going to print it and pass it out. And then when we get to the end of it, there's a couple of things that I did not do. So we were supposed to do composition of blood and white cell study. We did blood typing. We just went right by composition of blood and white cell study. And we didn't do the bacteriological examination of water at all, and we need to do that. We're going to do that tonight, and then I'll turn you loose. You can look at the example questions. I'll try to set up some more questions. White blood cell examples are over here. We've got a basophil. We're good.
Are these going to be the slides that are on the test, or?
I don't intend to change them, and they've been up for everybody, so hopefully they won't. Warren put pieces of tape on there and put the numbers so that if somebody moves them, we can find them again. Okay. So those are probably them. The ones that
Yeah, because sometimes they look different, like
right.
different
That
slides.
basophil is a really nice basophil.
Ooh,
It is so
I want to see it.